heavy metals affect plants. How plants manage heavy metal exposure. Heavy metals and depleted uranium are significant environmental contaminants that can adversely affect plant development and growth through various biochemical and physiological pathways. Their impact is mediated through various mechanisms, including toxicity, soil chemistry, and plant physiological responses. Heavy metals can disrupt essential physiological processes in plants, leading to reduced photosynthesis, stunted growth, and delayed development. This occurs due to interference with nutrient uptake and enzyme activity. Plants respond to heavy metal stress by activating detoxification mechanisms. These include demobilization of metals through chelation by organic compounds, compartmentalization in vacuoles, and the synthesis of protective molecules like phytochelatins and metallothioneins. Soil factors and soil properties such as pH significantly influence the bioavailability of heavy metals. For instance, lower pH levels increase metal solubility, enhancing their uptake by plants. Conversely, high pH can lead to the formation of insoluble metal compounds that are less available for plant uptake. Exposure to heavy metals can lead to oxidative stress in plants, damaging cellular structures and impairing growth. This stress is often mitigated by the activation of antioxidant defense system. Depleted uranium contamination can occur from military applications or industrial processes, leading to potential uptake by plants through contaminated soil or water. The uptake of depleted uranium by plants is influenced by its chemical form in the soil. The uranyl ion is the most soluble and bioavailable form for plant absorption. Soil pH and composition also play critical roles in determining the mobility and availability of depleted uranium for plant uptake. Research indicates that depleted uranium can accumulate in plant tissues, particularly under conditions where soil pH is low or where chelating agents are present that enhance solubility. Similar to other heavy metals, depleted uranium exposure can lead to reduced growth rates and developmental delays in plants. The mechanisms include interference with nutrient absorption and physiological stress responses triggered by toxic exposure. Long-term exposure to depleted uranium may also pose risks through bioaccumulation in the food chain, potentially affecting herbivores and higher trophic levels. Heavy metals induce a number of morphological and physiological changes in plants. Seed germination rate is decreased. The growth and development of seedlings is inhibited. In grown plants, leaves become smaller, wilted, and chlorotic. Leaf chlorosis and necrosis are apparent. There is a decrease in cell division in number of cells in the leaves, and the photosynthesis apparatus and photosynthesis itself are affected. The number of and size of fruit is decreased, and this is where there is a potential of accumulation of heavy metals. Stem growth is decreased, as is water and nutrient translocation to the above-ground plant parts. The growth of fruit is decreased. The number of secondary roots is decreased. The development of lateral roots is inhibited, and the absorption of minerals by roots is affected. Understanding biochemical indicators of plant well-being is crucial for assessing how plants respond to various environmental stresses. These indicators can provide insights into the physiological and biochemical state of plants, allowing for early detection of stress and informing management practices to enhance plant health. Among key biochemical indicators are phytohormones, such as abscisic acid. This phytohormone plays a significant role in plant response to abiotic stresses, such as drought and salinity. Increased ABA levels are associated with enhanced stress tolerance, influencing processes like stomatal closure and root elongation to conserve water. However, during biotic stress, high ABA concentrations can sometimes make plants more susceptible to pathogens by inhibiting other defense mechanisms. Osmolytes are another important indicators. Compounds like proline, glycine, betaine, and soluble sugars act as osmoprotectants that help maintain cellular integrity under stress conditions. They prevent cellular deterioration by stabilizing proteins and membranes, thus aiding in the plant's survival during adverse conditions. Antioxidant enzymes are also important indicators. Plants produce various antioxidant enzymes such as superoxide dismutase, catalase, peroxidase in response to oxidative stress caused by environmental factors. These enzymes help mitigate damage from reactive oxygen species, which can accumulate due to stress. Aquaporins are transmembrane proteins that facilitate water transport across cell membranes and are crucial for maintaining water homeostasis in plants under stress conditions like drought and salinity. Their expression levels can serve as indicators of a plant's ability to manage water efficiently. Specific proteins and enzymes, including those involved in metabolic pathways, can indicate plant health. For instance, the activity of hydrolytic enzymes such as phosphatase is often measured to assess soil health and its impact on plant well-being. Soil health is intrinsically linked to plant well-being, as healthy soil provides essential nutrients and a conducive environment for root development. Key biological indicators include microbial diversity. A diverse microbial community in the soil enhances nutrient cycling and disease suppression, directly benefiting plant health. Microbial respiration is an important measure of soil health. This measures the metabolic activity of soil microorganisms and indicates soil fertility and health, which influences plant growth. The presence and activity levels of specific soil enzymes can reflect the biochemical processes occurring within the soil ecosystem that support plant life. When depleted uranium contaminates soil, plants can absorb it, leading to potential bioaccumulation and ecological concerns. However, certain plants exhibit mechanisms that may help mitigate the harmful effects of depleted uranium exposure. Understanding these mechanisms not only highlights the resilience of certain plant species, but also opens avenues for developing phytoremediation strategies to address depleted uranium contamination in affected environments. Plants can absorb uranium from contaminated soils and water, where it can accumulate in various parts. The extent of this uptake is influenced by several factors, including soil chemistry, plant species, and the presence of mycorrhizal fungi. Certain indigenous plants have adapted to thrive in uranium-rich environment, suggesting that they possess specific traits that allow them to tolerate or even sequester uranium effectively. The bioavailability of depleted uranium is influenced by its solubility and the physicochemical properties of the soil. While plants uptake depleted uranium, they also employ strategies to limit its harmful effects, primarily through the regulation of reactive oxygen species and antioxidant responses. Exposure to depleted uranium can lead to oxidative stress, characterized by an imbalance between reactive oxygen species production and the plant's ability to detoxify these reactive molecules. To counteract this, plants enhance their antioxidant systems. Antioxidant enzymes are key enzymes such as superoxide dismutase, catalase, and peroxidase are activated to scavenge excessive loss. These enzymes convert harmful reactive oxygen species into less toxic molecules, thereby protecting cellular structures from damage. Plants also utilize low molecular weight antioxidants like glutathione and ascorbic acid. These compounds play a crucial role in neutralizing free radicals and reducing oxidative damage caused by depleted uranium exposure. Upon exposure to depleted uranium, plants may activate specific stress response pathways that enhance their resilience. The presence of depleted uranium can trigger changes in gene expression related to stress response, leading to increased production of antioxidants and other protective compounds. Studies show that certain plants exhibit adaptive responses over time, adjusting their antioxidant enzyme activities based on the duration and the intensity of depleted uranium exposure. For instance, superoxide dismutase activity may initially decrease, but significantly increase after prolonged exposure, indicating a robust adaptive mechanism. Some plants can produce chelating agents that bind to heavy metals like depleted uranium, facilitating their detoxification and reducing bioavailability within the plant system. This process helps minimize the potential for depleted uranium-induced toxicity. Mycorrhizal fungi form symbiotic relationships with plant roots, enhancing nutrient uptake and potentially affecting uranium absorption. Studies indicate that arbuscular mycorrhizal fungi can increase uranium accumulation in plants like Medicago truncatula and Pteris vittata, suggesting a bioremediation potential. Some plants are capable of phytoextraction, a process where they absorb contaminants from the soil and concentrate them in their tissues. This process can be enhanced by biodegradable amendments to the soil, which improve plant growth and uranium uptake. For example, Indian mustard, Brassica junca, has been studied for its effectiveness in extracting heavy metals, including uranium, from contaminated soils. Different plant species exhibit varying capacities for uranium uptake. Research has shown that certain crops and native plants can accumulate higher concentrations of uranium without exhibiting signs of toxicity, indicating potential for use in phytoremediation strategies. The chemical form of uranium in the soil significantly affects its bioavailability to plants. Factors such as pH, organic matter content, and the presence of competing ions can influence how much uranium is taken up by plants. While direct ingestion of contaminated soil is a concern for herbivores and humans, studies suggest that the transfer factor from soil to pasture grass is relatively low, indicating limited bioaccumulation through the food chain. A recent study by Riazudin et al. 2021 gave an overview of the heavy metals toxicity and the ways how plants manage to mitigate these effects. Thank you for your attention.